induction program. Now, we know that it is our neonatal Department of Zoology, Postgraduate Department of Zoology of Ramakrishna Mission Vivekananda Centenary College, Rahura. Now, it is we are immensely fortunate, and it is our very great pleasure moment for all of us that today we have with us Professor Dr. Subeer S. Majumdar, Director of National Institute of Animal Biotechnology, Hyderabad. Welcome you, sir. Now, I would like to request our respected and beloved Principal Maharaj, Swami Kamadasthanandaji, for the official welcome address. Maharaj, please.
So, most respected Professor Subeer Majumdar, our very beloved and very respected person, Director of National Institute of Animal Biotechnology, Hyderabad. He is our today's distinguished speaker, and my dear colleagues and my dear students of all postgraduate and BSc students, those are actually viewing from YouTube link also. So it is a very joyous opportunity for us, because amidst us Dr. and Professor Subeer Majumdar, very distinguished scientist in animal biotechnology, and he is also now the director of the only Institute of Animal Biotechnology in India, that is National Institute of Animal Biotechnology in Hyderabad. And he all through more than 20 years he has in-depth study and research in this animal biotechnology, and we are very fortunate to have in this beautiful evening for this induction program of newly admitted postgraduate students of Zoology department. So on behalf of the department, on behalf of all the students and faculty member, I extend our heartiest thanks and regard to Professor Subeer Majumdar for his kind consent and giving his valuable time for us in this beautiful evening. And I also tell the student, all you wholeheartedly participate in this program, especially the lecture of the sir, and he's very informative, very interactive person. So after the talk, you ask question, interact with him, so and learn lot of things, because he's something different person. So in his talk, you can understand how he is a teacher also. So with these words, I once again extend my heartiest thanks and regard to Professor Subeer Majumdar on behalf of this college. And thanks all of you for joining in this beautiful evening in this webinar. Thanks all of you. Thank you.
So, thank you, Maharaj, for your welcome address. Now, we know that it's a very usual that the Professor Dr. Subeer S. Majumdar is an eminent scientist of our nation. And we also know that no formal introduction will be needed for him. Yet, I am now requesting our presiding head of the department and chairman of the IQSC of our college, Dr. Pradut Kumar Midda, to say something about Dr. Professor S. Majumdar. Professor Midda. Professor Midda, please.
Dr. Subeer Majumdar is presently the director of National Institute of Animal Biotechnology, that is NIAB, Hyderabad. He has worked with non-human primates to obtain his PhD degree from National Institute of Health and Family Welfare. He did his postdoctoral training at the Southern Illinois University at Carbondale and University of Pittsburgh, USA. He initially worked on various aspects of male infertility using animal models, and later developed a few novel techniques to generate transgenic animals by manipulating male germ cells. He has developed techniques to generate therapeutic proteins in animal milk using gene transfection directly in the udder gland. His areas of specialization is male infertility, transgenesis, livestock genomics, and therapeutic protein production. The member of all three scientific academies of the country. He is recipient of Tata Innovation Fellowship and also of J.C. Bose Fellowship. That's all about Dr. Subeer Majumdar.
So, thank you, Dr. Pradut Kumar Midda, for your wonderful presentation about our chief guest, Dr. S. Majumdar. So now is the time ultimately comes for us, and now I am requesting our respected guest, Professor Dr. S. Majumdar, Director of NIIB, to deliver his speech.
Thank you, Maharaj Kamalastananda Sahib, and all colleagues of the college and all students who have joined recently. My very good wishes to all of you, and I will try to give a lecture where you try to understand that whatever you have chosen to join this program in the college, what is the attractive thing in that, what is future of that, and probably several people might have advised you to do what you are doing, but probably you will have a different look, different vision, because you were seeing it from the other side of the table, that studying zoology, what to do in future, those kind of questions. Today probably I will show you certain things on the other side of the table, and you may get impressed. That's my duty, to realize that whatever you have joined or you have stepped in is not a wrong choice, but it's a appropriate choice. Probably I will give you certain ingredients also to, so for cooking material, for example, everybody can buy potatoes and brinjals or egg and masalas, but everybody cannot cook a good curry. So I will probably be directly or indirectly teaching you how to make a good curry out of the ingredients of life. And that's what you have to learn. So a teacher teaches, but if the receptive brain is not that receptive, that teaching goes waste. But if the receptive brain is really receptive, that teaching can really make wonders. So that's up to you, how much you will learn, how much you will understand. Stay focused and you will enjoy it, because I usually do not give lectures to teach science only. I give lectures to make science attractive, science a thing of enjoyment, and science a thing which should ideally charm you to continue with. So I will start sharing my slides. I hope it shares nicely. Is the slide shared now?
Yes.
Can you see it nicely?
Yes, sir.
Good. Now I'll go to the first slide. I know. I'll do that. Don't worry. Now you can see it clearly?
Yes, sir.
Fine. So I will teach you. बीच में आ जाना. I'll teach you about life, genes, biotechnology, and needs of human. So before I begin, I would like to thank some of my students who have really helped me in generating information data throughout my life. Some of them, I am putting some of them, and they are Indrashish Bhattacharya, Sauvik Sen Sharma, Suvira Dhoop, Nirja Wadhwa, Dr. P. Nagarajan, he is a veterinarian, Abul Usmani, and Nirmala Ganguly. So Nirmala works with me here, but he is busy. So Maharaj Sahib was asking, or he sent him the link, but I don't know whether he can join or not. Presently, I work at National Institute of Animal Biotechnology, and earlier I used to work at National Institute of Immunology, which is in Delhi, and this institute I work today, it is in Hyderabad. So let us begin with science. So science, usually people join with a viewpoint of thinking that they will do research in their life, mostly. So let us begin with the topic of research. Now, why should I do that? Why should I do research? There are various viewpoints, there are various philosophies, parents, teaching, friends, colleagues, whoever gives you whatever guidance, based on that, you choose to do research. Now, if you ask me, why should you do? You just passed, you just graduated and joined a postgraduate program. So obviously, it's not that you are completely naive. You know what you want, and probably you have done what you wanted to do. Probably some of you have done because you didn't get any other option to do. But please understand, doesn't matter what you do. When you are in the water, you must learn swimming. Like that, when you are in this course, you must learn what to get best out of it. That's what I'm going to tell you. So the question comes, why should I do research? So I think this is our inherent character to search for something. We are born from childhood with this criteria to search, always curious. Even a small boy is curious and he tries to find it more magnified way. So what a normal person looks, a researcher tries to magnify and go very close and look into that. That's a researcher. Otherwise, every human being is a scientist. Otherwise. Now, why should you do that? I would say you should do it either because of curiosity, or to get name, or to get fame, or to do contribution to the society, or to contribute to society and feel dignified, to attain dignity. So most of the parents teach their kids that you should be a dignified person on this earth when you're born. One way to serve society is to become a medical doctor, or to become a veterinary doctor, or to become a researcher and try to solve the problems of the human beings, animals, any living being, plants on this earth. So I think these are the reasons why should you choose to do research, in my view. So it's up to you what you want. If you want to do take name and fame, people can have this frame of mind. Even then, they should conduct a good research and become very famous. Some of them can say, no. My parents taught me to contribute to the society. You can do that and really feel dignified. So this is why you, after you complete your post-graduation, after 2 years, should really decide to do research or contribute towards society. Now, what is research? In research, what is important, most of the things people have done. What you're trying to do is what is not done. For that, what is most important is innovation. So you have to be very, very innovative. You should think new. You should think a idea should be generated that should be a great idea. And you have to also think, does it meet the needs of today? That is, does it meet the modern needs? Because most of the needs people have thought very well and found answers. Anybody who has a brain can do research, in my view. But everybody doesn't do research. A man has to be thoughtful, a man has to be a great thinker, and the best research which you might have learned in very young childhood in class 3 or 4, the simplest research was that of a crow. That there was a water at the bottom of this pot and the crow wanted to drink it. No way it could put his head inside this and go to the bottom. That was not feasible. So it collected a lot of pebbles and put inside so that the water level rises up and it can easily drink the water. I would think that he is also a researcher. You see, when animals can do such a good research, why not we human being do good kind of research? Now, I will, since I am in Animal Biotechnology Institute, I will try to show you that how zoology, that the courses you have optioned, is study of life and animals. Botany is that of plants. So if you, I will only focus on the zoology and animals and why animal studies can help human beings or society. There are a lot of research which are conducted today on large apes or monkeys. They are for several diseases of metabolic disorder, physiology, behavior, and the interaction, human to human interaction. So animal to animal interaction also you can study. There are animals like dogs and pigs where you can study them for several hormonal interaction, several diseases of liver, or in dogs, several cancers. There are a lot of studies where you can study using fish, the lifespan, and you can study how they learn swimming, how their muscles work. There's a small, very small C. elegans, which is really being studied today for aging research. And just they have shown in this small creature that if you eat a lot of sugar, for example, and if you really try to metabolize it and enjoy, your lifespan gets cut. See, if usually the lifespan of this creature is 7 days, if you withdraw sugar by 50, 60, 70% from its life increases and doubles up. Instead of 7, it can live for 14 years. So see, what a great research. If you wanted to do this research in human, whether the life increases or not, you have to wait for 60, 70 years to see whether life doubles or not. But in this small creature, in 7 days, you can find out that life has doubled from 7 to 14 days. Similarly, based models at today's world is mice and rats. You must have known that a lot of experiments of drugs, etc., are done on mice and rats. You will inject something and see what happens, what is the reaction, whether a drug is working or not working. So see, all animals are at today's date used as a model to understand human diseases and also to find out the treatments, because you cannot give drugs or vaccines to human beings directly, because they can solve a problem in the brain and can repair a brain, but that drug can cause a damage to the liver. And the man can die of the liver disease because of the drug, but his brain will become all right. So it's of no use if the life is not left, if there is any toxicity of the drug or if there is a toxicity of the vaccine. So you need animals to do a lot of scientific research so that you prevent the sufferings of human beings and also that of animals, because several diseases are common to human and animals. So it is not that we only torture them, we use them for their benefit. And you will be surprised to know that almost all the drugs used in human beings are also used for animals for their treatment. So human has brain, it works, it uses some animals to do experiment and gives back that drug which is developed to both mankind as well as to the animals. So that's the beauty. Now, let me go and take the simplest example. If a man has cancer and if you want to see some drug works on that cancer or not, you can take those cancerous cells, and there is something called as nude mice which does not reject the cancerous cell from the other body. Usually body rejects foreign things, but in nude mice has very less immunity, and it is to be kept in a very cozy environment. And you can inject the human tumor cells, they will go and settle in some area and make a tumor under the skin, for example, and you can see it. And then you can test the drug you have developed for cancer and see whether the size of the tumor reduces or not. If it reduces, you know that the tumor size can be reduced by the drug, let us say called as drug A, but at the same time, you can take out liver, kidney, brain, eyes of the mice and test whether the drug has any ill effect on the other parts of the mice or not. So it's a wonderful model, good model. Now, is it that all the rats and mice we can catch hold of and give injections of the drug and see whether it is working or not? it is not true, because that's the basic research which you can do. But if you want to do advanced research, there are problems. Now, let us take a problem. In normal mice cannot be infected with poliovirus. Suppose I want to study poliomyelitis, where the muscles of the leg become very big, weak, and person cannot walk. Can I take a mice and inject poliovirus and it will display all the symptoms of poliomyelitis? Will mice have difficulty in walking? Answer is it doesn't happen so. Why, when I inject that virus into mice, it doesn't show me symptoms of human? Because mice lack the cell surface molecule that in humans serve as the receptor for the virus. So this is the receptor of human beings, which is present in every cell of the human being, of a man. And this virus gets internalized through this receptor inside the cell and propagates into all the cells and infects human. But if you take a mice, what happened? When this virus was injected, you see there is no receptor on the surface of the cell of mice. So it will not infect. That's why we were not seeing poliomyelitis. So I said biotechnology. So I can fish out from the chromosome of man the gene which makes this receptor as a protein and can insert it into a mice and make a transgenic mice gene transferred from one individual, that is human, to the other individual, that is mice. And then this mice will express human receptor. And then this poliovirus will get internalized. And then you will find that this transgenic mice expressing human gene for poliovirus receptor can be infected. Now it can be infected with poliovirus and even develop paralysis and other pathological changes characteristic of the diseases in human. So you see, a mice can be used as a model, but it cannot be used as a model of all diseases. So therefore, the biotechnology comes is that you cut the receptor gene from human, insert it into the mouse, and every cell of mouse now gives you human receptor, which is capable of internalizing the virus into the cell, and you see a mice which is a model or a humanized model of poliomyelitis. And you can do your research there. You can see how poliomyelitis occurs, how you can treat it with various kind of drugs. So basically what I was telling, that from human, you can take the genes of diseases, and if human chromosome is violate, you can cut a piece of it that carries the gene. You can insert into the green chromosome of the mouse, and this can be internalized and integrated into this chromosome. And now this is a humanized mouse model. It will display all the characters of the disease which human had into the mouse, and you can carry on with your research, which is allowed in mouse but not allowed in human beings. Now, what is this technology is called as transgenic technology, transferring gene from one individual to the other technology. What is this and how does it help? So genetic basis of human and animal diseases are very, very important. And if you know that, you can develop a strategy for various therapies. So you can generate these mice and study the functions of the genes. And that is called as, after we know all the chromosomes, all the genes which are carried by chromosomes, our job is, what do they do? What is the function of that gene? We do not know. A machine can give out a lot of information about genes. They will give you sequences of the genes, but machine cannot give you function of the gene. For function, you have to insert that portion of chromosome into the mouse and then see what mouse does. Suppose I insert a piece of DNA from human, insert in mice, and all the hair is lost. So that is the gene which is responsible for baldness, for example. And if you know how to stop that gene, you will be a millionaire, because you can sell that product in market and several people are waiting to buy that product to get the hair grown on their head. So now there are several diseases which are uncommon in animals. For that, you can generate transgenic mice, and they are called as the disease models in mice. You can also do few more things with the transgenesis. You can increase the body growth using growth hormone. For example, a farmer earns from a goat 13 to 14 kg of meat. You can generate growth hormone from human or from animal, and insert it and overexpress in it, and then per goat, you can increase the body weight by 2 to 3 kgs. You can increase the wool production from the sheep. So if a sheep is giving every season 4 kgs of wool, now it can overproduce it and give 6 kgs of wool from the same sheep. So that's a very, very big advantage. You can generate lot of proteins in the animals, lot of proteins. When I come to that topic, I'll tell you why those proteins are important and needed. So we'll come and go back to basics, what you have learned in your class 12 or BSc. So there is a cell, there is a nucleus, and in nucleus there are a lot of chromosomes. And if you unwind chromosome, they say that chromosome contains thousands of genes. A machine reads that there is a starting site and there is a termination site, and they say this is called as one gene, and machine will give you the sequence. That's how machine can read that there are 23,924 genes. But if you ask machine, okay, what gene number 16,920th does, machine will say, I'm blind, I'm a machine. I only give you information about gene. I cannot give you any information about the function of the gene. So remember, for your understanding, that the machine finds out the sequences which is called as promoter, and then the gene, and then machine finds out the sequences which is called as the termination codon, and says gene, one gene, gene number 2, gene number 3, 4, like that it will count and give you, all the sequences are different from each other. Now, for your understanding, it took a lot of time for me to understand the gene very well. And I was very frustrated. And you see, we were educated at a time when, DNA, RNA, that's it. No PCR, no advanced technologies, no real-time PCR, no modern biotechnology. And it took a lot of time for us to understand. So it also depends on who were your teachers. How did they teach you about a gene? So I have made a design for your understanding that every gene has to have something called as a promoter. That's like an engine. So this engine's job is to start the gene. This is the gene. So if you see the whole thing, neither the engine nor the wagon called as termination codon is going to do any function. The job of this promoter or the engine is to drive the gene and express the gene. The only thing which is important will come out is of these sequences, which is called as a gene. And this is the promoter which is only driving this gene, and this sequence will produce mRNA. You know that. mRNA will produce protein, and you know that that protein will do some function. So for functional aspect, this is the only important thing. This gentleman called as promoter only promotes it, and this gentleman called as termination codon or guard's wagon conveys that that's it. We are one. We are one, and we call ourselves a gene where there's a promoter and there's a termination codon. So our job is to carry this. And I was saying that poliomyelitis virus human receptor. So this is the gene which I need to cut and paste in the mouse so that this gene is expressed in the mouse. But promoter is one which drives the gene. Remember that. That's very, very important. Without promoter, the gene will stay where it is. It will not function. So for your understanding, a machine has given us a promoter. It read and said promoter has started, gene has started. Then it kept on reading, several exons are there and several introns are there. And then there is a termination codon. It says, that's it. This is a gene. But what we do not know, where this gene will go, what will it do, where it will reach finally, Lucknow, Delhi, Kolkata, I don't know. So we need to understand this through biology. That is called as biology. So machine gives a gene, its sequence, and we have to understand the biology. So we need to cut it and paste it in a mice, for example, and see the function, because function can be seen only in the lives. So basically, we can have foreign genes inserted into mouse genome and then try to see how the characters of the mouse have changed. Whatever is the change, that is because of this foreign inserted gene into the chromosome of the native mice, for example. So now I will show you a slide where we routinely do this work. When I was in Delhi, these are the videos taken in my Delhi lab, where this is a mouse embryo. So mouse embryo is about to divide. and from a single cell where there is a female pronucleus, and after fertilization, the male or father's DNA has swollen and become a male pronucleus, and in male pronucleus is relatively bigger than the female. That is why we have gene in this small tube, and we call it an injecting needle. And this is the zona pellucida of the embryo, as you have studied, and this is the ulima. Hello. So gradually, our job is to insert this foreign gene in a very small fraction into the pronucleus. Very soon they are going to fuse, mother and father's chromosomes are going to fuse, and this is going to become a single nucleus. And in this single nucleus, if you have added the foreign gene, it will divide, and this will make billions of cells of the body. And that gene will be integrated in all the cells. So how we do it? We superovulated mice, generate tons of eggs, and we keep all the eggs here, and then one by one we will pick up the egg by the sucking needle and hold it. This is called as holding pipette, and this is called as injecting pipette. It has got a suspension of DNA of foreign origin. For example, poliomyelitis receptor gene of human, and it is inserted into mouse's genome. So that's how we do it, that we hold it, and then you can see this is male pronucleus, we will inject it. It will get injected, and this will swell because a small volume is going inside. You can see the swelling. Then this egg will go here. It's an injected egg. Then this pipette's job is to, our technical officer's job is to bring this pipette up, hold another egg, bring down and inject the gene. So when it goes up and brings another egg, you can see this female and male pronucleus, but one more sperm is trying to get in. You can see the sperm moving. Very interesting. Again, we will do the same procedure or same phenomenon and try to insert foreign gene. Like that, several eggs, or in a day, around 80 to 100 eggs can be injected like this. There is another phenomenon by which you can do transgenesis. That is, this is another work we have done and published. This is a buffalo oocyte. And in this, I am saying oocyte, it is not yet fertilized. So if I stain the chromosome material with hexed dye, it will give me staining of DNA. So there will be intense staining, which is called as polar body. We are not interested in that. But you can see a staining which is relatively less intense because this is like a football. And inside the football, that staining is there. So it is filtering out and its intensity is getting low. That is the native nuclear material of this egg. I want to remove this native nuclear material of this egg and throw it away. So this egg is now enucleated egg. It doesn't have its own chromosome or DNA. I can culture a lot of cells here and insert foreign DNA into those cells. 30% of those cells, for example, will have the foreign DNA. I will take out nucleus from this cultured cell, which has foreign DNA integrated, and push it inside the enucleated egg. So although cytoplasm and all the body of the egg is that of a buffalo, this is a buffalo egg, but the nuclear material is that which you have inserted from outside. The whole machinery will support for the growth of those chromosomes and display their characters. And we can have an animal model, which is like what this inserted nucleus with foreign gene is. This is called as cloning. If you have studied about Dolly, sheep, there the nuclear material of the original egg was thrown away, and from a mother, another nucleus was taken and thrown it inside. When this individual grew, that grew into Dolly, it was exact replica of its mother. This is called as cloning. But in that way, also you can insert foreign gene inside this nucleus and create a transgenic animal. So let us see how that is done. So you can see this is very intense, this is on the top, but inside football, actually this is the body inside the egg. That is removed. All the nuclear material will be removed. Is sucked into the tube and gone out. And then we take cultured cells which are transgenic, which has got foreign DNA, and we will insert this nucleus into this egg. What is the benefit? Now this egg has the genes or the characters I wanted. So I know exactly what I want, and this, when the egg develops, it is exactly what this cell was, and it can be For example, a sheep with great amount of wool, or a cow with great amount of milk, or a bull with elite sperm qualities, and now exact replica will be made. So I have got a ready-made same to same guy generated. That is also transgenesis. So for example, if we culture the oviductal epithelial cells of buffalo and try to transfect all of them with green fluorescence protein. Green fluorescence protein is a jellyfish protein which gives you green color. When I try to transfect, not all the cells are transfected. You here see only two cells get transfected. But I know that this nucleus of this cell, nucleus and cytoplasm, so this nucleus, the foreign DNA called as green fluorescence protein has integrated. So I can take out this nucleus and insert into that enucleated oocyte which I have just shown. Ideally, I should get a green embryo. Usually, buffalo embryo is not green. But how do you see it? This is called as phase contrast microscopy. This is in light. But you can turn off the light and turn on the UV. Then only the fluorescent material is seen. So all the adjacent cells which did not integrate GFP are not seen here. You can see here. But only two cells which integrated GFP are fluorescing. These cells are not fluorescing, right? So if you take this nucleus and insert into the egg, you can find out of the two, when I turn off the light and turn on the UV, I see this is inner cell mass which makes the embryo, and this is the trophoblast which attaches to the womb of mother and makes the umbilical cord. If you have studied, you're a zoology student, will understand this. So you see, this egg didn't get my gene inserted properly. Both of them are here. I just turned off the light and turned on the UV. And only one is showing green. Out of these three, only one is green. So successfully, you can insert foreign gene by this method. But these are very, 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 very difficult methods. I have just shown that how to do that. But this method is actually very difficult. You have to need thousands of mice eggs at the cost of 100 of females which you will kill and take out those eggs, and or cloning, which is very, very difficult. It is not easy. A technician needs 1.5 to 2 years to learn the technique. Really, only then it will get a certificate for only one technique. Very difficult. So it is not so easy that every lab in the world can do it. It's a very specialized technique and done in very few places. So this is really bothering that we cannot produce fast, very rapidly, several animals with the genes of our interest. This is difficult. Now, I'll tell you that why the egg was used, because there was a lot of gene pairing, breaking, going to happen at the time of cell division, and crossing over. That occurs at the time of cell division. Lot of genetic material even between chromosomes can be exchanged. So we know that this kind of phenomenon occurs only when the cell is dividing rapidly. That's why original thinker or researcher added a foreign gene here, that now single cell is going to become billion cells and my gene will get integrated very quickly there. If I use a foreign gene, and in the hodgepodge situation, this can get integrated. Now we started thinking that if embryo technique is so difficult, and if you go to large animals, you have to give large number of cows or goats or female buffalo to get superovulated egg. In mice, you get 30 egg. But if you want to make a large animal transgenesis, buffalo, only 2 to 3 eggs after giving hormone injection. Normally, 1 egg. Mice, 12 babies, 10 babies. Normally, if you give hormones, 30 eggs. So large animal, it is next to impossible. You can't do it because you have to have a big herd of 100, 200 cows. Who will pay for their food? It's very, very costly. So we started thinking, can we find out an alternate way? So I'm trying to give you an idea that how you have to be a thinker. So in my lab at NII, I used to do this work. A lot of requests will come throughout India. And it is very difficult to fetch to their needs. Out of 70, 80 people requesting me, I could do this in a year for only 10, 12 people. So I thought, why not to design an experiment or discover a method which is much easier, which can be done very fast, for which you don't have to wait for a long time, for which you do not have to sacrifice so many animals. So it is very, very important to know that if just by transferring gene and we could get few transgenic animals. So whenever you transfer these eggs, now these eggs are to be transferred into a pseudo-pregnant mother. The eggs I have shown you, which you have manipulated, where I have inserted foreign gene like this. We transfer it into a surrogate mother. So mother is hormonally prepared to carry a baby, but it doesn't have its own embryo. Then we transfer these embryos. And if this is a foreign gene, then after a baby is born, as I said, every cell of the baby should have that foreign. So we cut a small piece of tail and do something called as southern blotting, or we do PCR and try to see that which of the tails of 10 or 12 babies born have my gene which I have inserted. And we find that only 2 of them have. In southern blot or PCR, you will see amplification in only 2. So 1, number 2, 3, 4, 5, 6, 7, number 8. So 2 and 8 are transgenic. Rest of them, God did not help us to make them. So we'll throw away these animals and keep these animals and use them as transgenic animals. So you understand, you have to have a set of animals for superovulation. You have to have a very good technician trained for 1.5 years for transfer of gene. You have to prepare surrogate mother. Then several babies will be born, few will be only transgenic. And all these animals are killed for superovulation. As I said, sometimes 100 animals. So this technique of inserting foreign gene is very good, but success rate is only 1 to 8%. In India, first transgenic mice was made in National Institute of Immunology, where I used to work before. And this was, that something is called as hepatitis. So that happens because there is a gene called as HBVX gene, hepatitis B virus. Its X gene, the gene is coded as X. If that gets inserted into liver cell, the liver enlarges. So we generated transgenic mice there at NII, and the mouse from the top would look like this, bulged tummy because of the large liver, and normal mice would look like that. And as I said, you can cut the tail piece and measure. That's the reason the tail is cut. And when we found this is transgenic, you see when the liver grows, it displays the characters of liver cancer. It's a big liver, and whereas control liver is small liver. So now you have a model of transgenic mice. These mice can keep mating and producing hundreds of babies, which will be displaying the characters of liver cancer. So you have got a model where you can study liver cancer. But you have to think that this technology is very difficult. Everybody cannot do. In India, only there are 2 labs which do it routinely. Now there are another 2 labs, but earlier there were no labs, so not so many labs. So as I said, you have to, first thing is you have to generate new ideas. For that, you should always sit and think what is to be done, how it can be made easy, how it should not be 2 or 3 labs, but how it should be done by every lab in India, how it can be very, very easy. So there should be a solution to every problem, and you have to find the key. So our lab started thinking what to do. And we thought, I'll give you a very good example, that suppose I am the foreign gene, and one fine night, I go to a place where I am driving my car by the lane, and my car gets punctured. I take out my cell phone, try to call the mechanic, phone doesn't have any connectivity. So I get very frustrated. I go and knock the door of a person, and there I see 2 friends are sitting in their drawing room and enjoying their life. And I try to push the bell. They open the door and they say, "What?" I said, Sir, I wanted to use your phone. And he just gets very annoyed. And what he does, he tries to shoot me. And then I said, why he's getting angry? Then I said, no, I can also have a drink with you. Please understand, listen to me. No, he just takes out his pistol and is about to kill me. I run away from my life. I really ran away from my life. What happens then? That's my office route. So after another 6 months, I am walking by that route, and I find that there is a wonderful bhangla going on, and the whole road is blocked. There is no way that I can take my car. So I park my car on the side and I wait there. They are not moving. They are moving very slowly. So I wanted to go back by walking and see how long is this procession and when can I go home. What I discovered, the same gentleman that evening who was sitting, his brother's marriage is probably going on. And he watches me and he immediately recognizes my face, because that day he saw me very closely. And he might have also repented later on that why was I trying to shoot that guy. But that day he recognizes me, and probably he's little drunk. He comes and embraces me and says, "Oh, come on. तुसी कैसे हो यार? And I said, sir, I am fine because I am worried he has got a gun. I know he has got a gun. So I said, no, sir, मैं ठीक हूँ, तुसी दसो, तुसी कैसे हो? Then he says, तुसी हरविंदर दे शादी दे वास्ते आए हो? Then I understand that Harvinder must be his relative sitting on the ghoda or ghodi. I said, I am fine, don't worry. He said, no. Tanu nachna payega. And I said, no, sir, मैं नहीं नाचता. No, तुसी तो मेरे संग आओ. He brings me to the side of the road, opens the dicky. In Delhi, this is the style. And there are some drinks kept. He serves me drink, he forces me to drink and forces me to dance with him. I am very afraid. I can't say no because he will shoot me. I know he has a gun. I was very shaken. After half an hour, I go to him, say, sir, can I go my home? My car is also left far behind now. Procession, I have danced with you. No, you cannot go. You are my family member. You have to come and attend the marriage. You have to go to girl's place. Again, I am afraid he has got a gun. I go to girl's place. Marriage occurs around 11 o'clock. I say to my wife that I am stuck. Wait, I'll come late. At 11 o'clock, I say, sir, can I go? He said, no. Tomorrow morning, there will be Vidai. The daughter will come to our house. You have to come to our house. You have eaten in the girl's house. You have to come to my house and eat. Whole night he retains me with them, brings me to his house next morning, and then feeds me and leaves me at around 11, 12 o'clock in the afternoon. What I am trying to tell you, the same gentleman, when he was sitting quiet on an evening, did not allow me to get integrated. And one day, when everything is haywire, everything was haywire, I was easily accepted by him and intermingled in the whole family. So let us consider that this is a chromosome, and when a single gene is going to integrate in a chromosome which is sitting idle like that evening, it is not allowed. But on a day when the chromosome is dividing and there are thousands of divisions occurring, everything is haywire, same gene is easily integrated very nicely and happily within this system. So we thought that embryo divides. At that time, thinkers put gene and made transgenic animals. The foreign gene got integrated into the native genome and a transgenic mice was born. In testes, spermatogenesis occurs, and you will be surprised to know that human produces 200 million sperm per day. Think. That means huge amount of mitosis and meiosis is going on in the testes, just like what happened immediately after fertilization, embryogenesis, tons of mitosis, meiosis occur. So, we thought that instead of going to oocyte, why not we use testes where germ cells keep on dividing rapidly in millions. And if we insert gene, probably like this system, my gene will also get integrated. So existing technique uses females 100 to 200. We have to sacrifice to get egg. And we get one founder animal, orange colored I show you, or two founder animals. But if we can go to male, use its testes, male is alive, you are only using testes, inserting gene, and then you have to give a small current. When you give current, the germ cell's mouth opens and the solution of DNA goes in. And because it is dividing or bhangla is going on, the gene can get integrated. So we did that. And that is what is biotechnology, that we have taken a gene from some other organism and we have done a surgery and we have inserted that gene in the testes and we have really given it by a small electric current. And there, when we wanted to do the same thing to both the testes, the animals used to die. So we started doing in one testis and we would surgically remove this testis and throw it away and put everything back and suture. This mice has now one testis where foreign DNA is inserted and we put this mice for mating. And what we found that if this is the testis, if I cut the testis one half and throw it from the top and try to see with a staircase from the top, testis would look like this. There are a lot of seminiferous tubules. These are the tubes which come up. These are the tubes which are going up. And these are the seminiferous tubule boundary. If our gene is inserted, there are some stem cells here. See, stem cells' job is to divide and divide more, but stay stem. Divide and stay stem. So that is why for 70, 80 years of age, a man can produce or keep producing sperm. If I can hit with my technique, some of the stem cells, whole life they will produce transgenic sperm. We did that, and we found that if we insert green fluorescence protein, which is only for expressing green fluorescence, just to test whether the thing is working or not. So one test is we did not electroporate the gene, non-electroporated test is. Another test is we electroporated gene, and we found this test is not express any GFP, but this test is where gene was inserted is expressing a GFP. So you turn off the light, turn on UV, you are seeing a lot of GFP. So it appeared that this technique will work. With that, this is the small portion of the seminiferous tubule, and these are the stem cells. And our job was to get to the stem cell. This is a seminiferous tubule boundary. Here there will be another seminiferous boundary. Here there will be another seminiferous tubule. So it is just like a pile of sugarcane sticks kept on the side of a room. And in between those sticks is a material which is a cementing material. So we wanted to hit this stem cell. If we do that, for rest of life, it will make a transgenic sperm for me. Beauty is, I'm not killing a lot of females to get egg. This male, if I put it for mating with the females, this transgenic sperm will produce transgenic babies. We did that, and we have successfully produced this. And somebody, if you have studied, you will understand that this is a diagram which is showing that if this is the testis which is electroporated, 13 number male for us, 13th time we were very successful. And these are the wild type 3 females which were mated. They all produced progeny. One of the females, none of them were transgenic. Another female, 1, 2, 3, 4, 5 babies were born. 3 were transgenic, 2 were not. Another female, 1, 2, 3, 4, 5 babies were born. 2 were transgenic. And then from transgenic, when we again mated with wild type, we produced more transgenic animals. The green ones are transgenic. So we successfully did it. And this really is an extraordinary output of good thinking. And now this is a very big journal called as Nature Methods, and we publish this in the Nature group of journals. It's a very big journal. And you can confirm by doing Southern blotting that you can insert the foreign gene. These are the foreign genes which inserted in several places into the chromosome. So you don't need to understand this. You have to grow up to know what that. But I will only say that this is one kind of transgenic baby. When its second generation baby was born, it gave the same kind because this band is present here. This is another kind of transgenic baby where this band was missing. When it produced its baby, again you can see it is just like its father, that it does not have this band, which proved that not only we generated transgenic animals with foreign genes inserted, but they also propagated to next generations. That's the importance of transgenic animal, that it should propagate from generations to generations. So you have done a permanent integration. So for example, there was a thread of red color in the native genome. You have inserted in several places small blue colored pieces of threads. And from babies to babies to babies to baby, red with some blue threads have gone. So you have got a line of transgenic animals with which you can do any kind of science you want to do. Don't worry about all these things. But with that, when we wanted to publish in a very big journal, they said show that it can cause a change in some system or some organ. So I have told you and shown you the engine, that's promoter, and the tube, that was the gene. So beauty of the promoter is that promoter is tissue specific. All our body cells have same chromosomes, but only in brain, certain genes are expressed because the promoter or the engine is working there. In liver, for enzymes, certain other genes are expressed because promoter or the engine for that is working in liver. If the engine which is working in liver starts working in brain, my brain will produce all enzymes for digestion. I don't want that. My brain will be digested, right? So the beauty that promoter or the engine is tissue specific. So we generated a mice which had a tissue specific promoter of brain and tagged something there, and we found that in normal mice, you can see this is called as glial fibrillary acidic protein. Glial fibrillary acidic protein. So GFAP is very nice in control mice brain. So this is a tissue section of the brain. But when we generated a pathological model using something called as human IGF-BP overexpression model, that will express only in the brain, and it will disallow the production of human glial fibrillary acidic protein. You can see it is diminished here. Same mice, same species of animal, in one it is controlled and one it is transgenic. Then this paper was accepted in a big journal. It's just for your showing. Now, what we have done, that instead of oocyte mediated transgenesis, which is very cumbersome, technically very challenging, very time consuming, we generated transgenesis via testicular germ cells, and that was developed at NIA and published in Nature Methods, where we give small electric current to germ cell, its mouth opens for a while, and immediately the foreign DNA goes in and integrates. So what we have done is we have generated an easily adaptable technique for rapidly generating humanized animal models in the era of human genome. So this is the era of human genome. Good machines have come, and very fast they can tell you about genes, but what they cannot do, very fast they cannot produce transgenic animals to know the function of that. It requires very few animals. It requires less time to develop. This is the take-home message. So this diagram is just showing the take-home message. Requires less time to develop transgenic mice, requires less time to study more genes in less time, does not require costly infrastructure or skilled manpower. Embryo manipulation laboratory is somewhere around 1.5 to 2 crore rupees. You don't need that laboratory. Just stick in a needle, give the gene in testes of mice, give small electric current, put it for mating after 30 days with females. It will churn out a lot of transgenic animals. And most importantly, it is ethically superior because it's deathless. You're not killing any mice. And you can use this in large animals also. You don't have to have a herd of 200 or 600 female cows or buffaloes who pays for it, for their food. So even one bull is good enough to do this electroporation. So this is a real picture. So what is the intensity of science? This is a real picture. And I still remember that there was a student of ours, and he was not my lab, somebody else's lab, and he was a very he had a very good camera. I borrowed his camera for 4 or 5 nights and tried to take a picture of a mice. This is actually a paper clip. I have made it straight. This is a plastic or whatever paper clip. I have made it straight. Then I put a thank you sign and took 3 nights to take a wonderful picture as if it is holding a mic and saying thank you. You saved lives of several of us by generating this technology. So this is the expression of our happiness of our team, and we did that sitting 3-4 nights, and we could capture really very good photograph. But this technique is still needed. Always think whatever you have done is not, it can be made better. It can be made more better. It can be made more better. When people tried to use this technique, the surgery business was not a piece of cake for everyone. Then there is a neurologist, there is a hepatologist, there is an endocrinologist, there is a ophthalmologist. When he wants to do his experiment, he may not be a good zoologist like us, who can do wonderful surgeries and neat and clean and stitch it back and put it back. While doing that, either they would harm some nerve, causing impotency, or they will do some other disturbances causing infection. And this technique was becoming really difficult for others to use who are not great surgeons for the small animals. So we developed a new technique, and what we did, that in others' hand it becomes so bloody. So we didn't like it. When you give to other students, they did like that. So we didn't like it. So later on, we worked very hard, and now we have developed a technique where you can insert genes into the both testes from outside, do not do surgery, and do electroporation, and you can generate transgenic animals. I'll give you a proof that we generated transgenic animals using new technology without any surgery and no blood, and we could still generate a transgenic mice where spleen size was increased as compared to control animal. This is a proof that we could generate that transgenic mice. So there is a SMAR1 is a protein. If you overexpress that in mice, the spleen enlarges. So we could do that as compared to control mice. We also did, one of my students, the Ganguly whom I was talking about, he is also crazy. He isolated his own keratin 14 promoter. Keratin is the upper layer of the skin. So I said, promoter's job is to drive the gene in a specific tissue. So keratin is upper layer of the skin. So he isolated promoter, which is expressed here, a protein called as keratin usually expresses. But we just detached the engine from keratin's protein gene and attached green fluorescence protein and made this transgenic mice. So when you turn off the light, this is the piece of skin, and you see the upper layer, which is keratin layer, is green, which clearly indicate that we could successfully generate transgenic mice using keratin 14 driven green fluorescence protein using a new technology, which is a bloodless technology. And this was also published in a journal called as Scientific Reports. So we have, instead of using oocytes and killing several females, now we use testis mediated transgenic, and we can generate several founders from one male which remains alive. It even doesn't die. That's a very beautiful technique which is developed. I am showing you to generate your interest in this kind of science. Now I'll go to other thing. Now let us understand that when a adult man is having a cancer, then you have to give chemotherapy, which is a drug, or radiation, which is a ray. So suppose this gentleman have cancer of liver, but when he is exposed to the drug for chemotherapy or given repeated radiations to kill the cancerous cells inside the liver, what happens? That cancer is a dividing cell. So when you give this treatment, their job is to kill all the dividing cells. That's why the cancer cells die. I told you the testis makes 200 million sperm per day. So in testis, all the germ cells are constantly dividing. When you give radiation or chemotherapy, the testicular germ cells are dividing. That's why they will die. Because chemotherapeutic agent or radiation doesn't or cannot dissociate between dividing cells cancer and dividing cells non-cancer. They just kill dividing cells. So during this treatment, this man can survive or after this treatment, but he acquires additional, I will not call it disease, I will call it deficiency, called as infertility. His sperm cannot be produced because all his germ cells have died. Now, if you come to adult male and give this treatment, intelligent people say, don't worry. Let us take ejaculate of this man and save it into liquid nitrogen and let him become infertile. We will do with this stored sperm, whenever he wants a baby, we can take superovulated egg from his wife and stored sperm and fertilize and transplant back into the female and produce a baby. Doesn't matter if he is infertile for the rest of life. The question came in, if this male is less than 12 to 13 years of age, he doesn't have his sperm, but he is suffering from a cancer. So when this small gentleman is given treatment of cancer, there is no sperm, but it's all germ cells die. Then what happens? It's a very difficult situation. So ideally, you should really, what do? I do not know. So this is a very critical question. But what you can do, that you can isolate these small germ cells, culture them, multiply them in large numbers. As soon as you culture, they will divide, and freeze them in liquid nitrogen. And once you freeze them in liquid nitrogen and give the chemotherapy, its germ cells, native germ cells which is residing, will die. But from a small fraction of the testis, all the germ cells which are preserved, after the boy grows up and becomes 13, 14 years old, you should be able to transfer back this germ cell into his evacuated testis, which is evacuated, germ cell evacuated because of the treatment of the cancer. That's possible. Now, to do that, first you have to check it in animal whether that's possible or not. So what we did, that we used one of these chemotherapeutic agent and injected directly into the testis. This testis became smaller in size. So when in an animal, we have not injected anything, we have just injected buffer. This is a mice, a good testis, nothing happened. And in another, we injected the chemotherapeutic agent and the testis became small. Same age, they are brothers, born on the same day to the one mother. But because of the chemotherapeutic agent, the testis of mice became very small because all germ cells have died. So it has shrunk. Its seminiferous tubule, which I was showing, is blank. Now, if I would have saved the germ cells from one of the testes in liquid nitrogen, then I have an option. If I give it back to the testes, does it grow and does it produce a baby? So we did those experiments, and I will show you a technique which we do again in our lab. See how beautiful and technology is. And there you will see this is the evacuated testis, and there is a blue colored solution in which the germ cells which were stored are being given back to him. There are a lot of seminiferous tubule in testis, and we are going from efferent duct, from where actually sperms come out, we are going reverse, and we are injecting into the shrunk testis the germ cells, but we give trypan blue solution to check whether it is going to the seminiferous tubules or not. You can see the tubules are getting filled up. Can you see? These tubules are getting filled up. These tubules are getting filled up. And they see wonderfully these tubules are getting filled up with solution because these tubules are vacant. They are waiting for germ cells. So we have inserted the frozen germ cells back to the testis. And this is a technique which is called as germ cell transplantation. And by doing so, you see how nicely seminiferous tubule get filled. We have successfully produced babies after we have inserted the germ cells. So this is a very good technique which we have done, and we are writing the paper to be submitted. It's a beautiful technique where cancer patients' germ cells can be stored, then cancer treatment can be given, and then after a year or so, those germ cells can be transplanted so that they go back and home, and the man does not become infertile, and he can father his own children. He doesn't have to take a donor for the germ cells. Although now, because the techniques of cancer treatment have become very, very good, you can really survive forever with good cancer treatment. But you do not have to survive with infertility. Good thing. Now I will switch gears here, and again I will come to gene and its manipulation. And I said you have to give something to society. So now let us see, there is something which society is needing today. That is called as therapeutic protein. Protein is a protein, you know. Our body made hundreds, thousands of protein. They do some or other function, and we are normal. Therapeutic protein means a protein required for treatment. That means when our body, either because of disease condition, cancer, arthritis, so many diseases, stops producing that protein, then we have bodily dysfunction. When my body stops producing that protein, so I have to exogenously inject that protein into my body to repair the loss, because now my body doesn't produce it. But how to insert that protein from exogenously into the body of a human being? That means you have to generate that therapeutic protein somewhere outside. So I'll just say, due to various diseases or aging, some proteins are produced less than required quantity in the body. These proteins are really needed to be replenished to the body, right? So we need to give the injection. See, human or animal proteins. Animals also have diseases. At present, animals also need hormones to regulate their fertility or gamete production. So these proteins are produced in vitro or in vivo and are used for these therapeutic purposes. In vivo means body. This is a new science. Mostly it is produced in vivo. So if you know that you can insert human genes into bacteria and can have large fermenters in big factories where bacteria will grow, along with that human gene will grow, and bacteria will produce this protein. Then it's a huge techniques to be used to extract those proteins from bacterial body and then purify and make injections. So that when you replenish, the body will become good. Production of these therapeutic proteins in large quantity with proper structure and biological activity is the need of the future. That's what we need. Tons of those bacteria are to be grown in big industries. So some of the example I will give you, Guillain-Barré syndrome is that a bacteria or virus can get inserted in your body. Your body will produce antibody against those bacteria or virus. Those bacteria and virus will die and go away. Sometimes body thinks that those antibodies think that their own nerve coverings, they think that this is the bacterial covering, because there is similarity between these coverings and the surface of the bacteria or virus. So the antibody now generated to kill virus is now acting on its own body and trying to distort the surface of the nerve. So conductance in the nerve goes and man can lie flat like this without any movement. Immediately you have to give lot of IgG injections, so good IgGs to compete out these bad antibodies, and the person can recover in a year or so. I have an example in my own institute in New Delhi where I have really seen a person all of a sudden lying flat like this, and it took a year for him to recover. So he used to get injections and each injection would cost around 11,000 rupees. 3 injections of IgG in a day, and the treatment went up to at least a month, and then gradually he recovered. There is disease called as arthritis, that is because of the inflammation. So BMP2, bone morphogenic protein 2 and 7 are used to treat such conditions in human. But you see, these treatments are luxury of very rich people. Poor people cannot get it. Thousands of rupees from where they will get for a while. It's not possible. Now, why cost is more? Because you need a very huge factory, 3 shifts of duty, electricity, license, purification of protein, so many things going on. Who pays for that? That small vial of medicine, which can be produced in less, but because of the way it is produced, the cost is very high. Factory cost, light cost, 3 shift duty cost, and licensing cost. So cost is exorbitantly high. So these are various therapeutic proteins here, and the cost is in the range of 13,000-14,000, and a set of treatment is 3.5 to 4 lakhs rupees. I do not think any rickshawala, tangawala, or a poor person can really afford it. Even a lower middle class person can't afford it. So because these huge factories are required, so think of the creation of the factory cost, crores, billions of rupees. And this way, these are the fermenters where these are grown. Who will give this? So one of the way to think in biotechnology is, we know transgenesis, we know promoter, which is a tissue specific promoter. So if you can isolate a promoter of a gene which actually functions in the udder gland, that means engine is that of a protein which will be definitely active in the udder gland, not in brain or liver. Behind that, you try to cut and paste or stitch human gene of interest. So that pipe I have shown you. So promoter, the engine is from the buffalo udder gland, and that pipe is of cut from the human gene of, let us say, bone morphogenic protein or IgG, coding for that, and they are tagged. Now, if I can create a transgenic animal like big like this, that promoter will only work, that engine will only work in udder gland, and along with this blue colored native milk, it will produce this red colored milk, which is because of the insertion of a gene from human. So human protein can come here. What is the beauty? This standing animal is my factory, is my construction, is my 3 shift duty, is my electricity, is my license. I have to only feed very less as compared to this expense, and the milk comes in very large amount. So you can isolate the protein, and cost from $13,000-14,000 can be made to, let us say, 100 to 120 or 150 rupees. So instead of 13,000-14,000 rupees, sorry, not dollars, it will be 120 or 150 rupees. So poor people can afford it. So that is our target. Then how do we work? So first we have to dig out the protein which is expressed in the milk. We have to finally take out the engine of that protein, that is called as fishing out promoter. We all know there is a protein in the milk called as beta casein. So you know milk casein. So we have isolated the promoter, the engine of the milk casein from the chromosome of buffalo. And if we do that and take a human piece of human gene and insert it under the promoter of udder gland protein beta casein in the milk, I should be able to get that. And as I said you, that instead of 14,000, our target is to make it 100 or 150 rupees per vial. So that's what we are trying to do. How we do that? We have taken and the chromosome of the buffalo and fished out the promoter and first 2 exons of the gene of buffalo, where this promoter will always drive anything tagged behind it, always in the milk, nowhere else. And I will not go into detail. Instead of milk based human protein, we tag enhanced green fluorescence protein. First, we have to track, can we, if you tag under this udder gland promoter a green fluorescence protein, is it that only udder gland becomes green? No other area becomes green? That's for visibility. Once that is confirmed, then we can, in place of EGFP, we can put human gene. So let us do that experiment, and what we eventually will do is we'll go to goat and do that experiment. But before that, what we have to understand that how it will work. So let us take an udder gland. There are milk ducts. Some of you students can ask me a question that when that protein is made in tons inside this gland, it can go into the blood. And suppose this protein usually is found in blood as 5 nanogram per ml, but if I am making it in milk gland, it can become, instead of 5 nanogram, 5 million nanogram per ml, and the animal will die. You are right. But it doesn't become that. Why? Because blood comes from here and flows from the surface of this gland. Blood never gets into the milk. You have never found blood into the milk. Whatever milk produces, a mother, a woman, or a cow producing beta casein inside this duct or a gland during the period when mother is feeding a baby, her blood level of beta casein doesn't go 500 million grams or milligrams. No, nothing goes from here to there, nothing comes from here to there as a leakage. This is called as exogenous gland, where whatever produced here is just externalized. The blood goes from here to here, and whatever you produce is just externalized. It never goes back. Taking advantage of that, we can insert something into this duct and transfect these cells, which are the udder gland cells. Then they will produce the foreign protein in this milk. What is the beauty? If I go into the back slide and show you this, let me say, nowadays, lot of people are saying that this is called as genetically modified organisms. And GMO is not acceptable. You might be reading that GMO is very difficult to accept. We have BT brinjal, BT cotton, now BT mustard. It's very difficult to accept. The government, the society does not accept. They are worried that since we have manipulated the whole body of this animal, maybe after 7 years, this animal will become mad. Maybe after 7 animals, this animal will have a foot and mouth disease very easily. That's long-term future. That fear doesn't allow to have the whole animal transgenic. So what we are trying to do, we are trying to just go and inject in the udder gland the foreign gene and make this udder gland only having foreign gene. So nothing will happen to brain, liver, lungs, anywhere. And we can have human protein from here. If somebody steals my buffalo, it cannot reproduce because I have not inserted this gene in every cell or egg or sperm. It cannot be reproduced. So that's the very, very good safety. That is called as non-GMO, non-genetically modified organism. So we are working towards that. And what we have done for that, we have fished out a promoter udder gland, and we are now injecting that gene with promoter of udder gland directly into the udder gland. So it will produce milk with human protein. It will be exogenized here. And that's a very important thinking. I again think you should be a good thinker if you want to become a good researcher. So this is a mice. So experiment is done in mice. We have inserted, this is a teat of a mice. So see how finely we have inserted needle into the teat of a mice. You will find that. And in this teat, we will insert a needle. You see? And then we'll push that gene. And only udder gland area will be having that gene, not the whole animal. And if you have really inserted this foreign gene, it should turn blue because I'm injecting. You see, foreign gene has gone there and only to udder duct. It doesn't leak anywhere. Then if I cut this area and try to see, is my gene gone? Then I'll find the gene is gone. Now, I'll tell you. Now, here, if you simply inject the gene, it will not get integrated. Today, the COVID-19 virus thing is going on. You know what virus does? It attaches to the membrane of the lung cell, fuses and releases its nucleic acid material into the lung cell, and thousands of viruses are made. So virus's job is use its surface, use the surface of football to attach to any human cell, fuse the cell membrane of human cell and release its own DNA or RNA material inside that cell. So we have taken advantage of that technique or biotechnology. Now what we have done? This is a Sendai virus. Ideally, it will have a membrane and it has got its own nucleic acid material. It's a viral nucleic acid material. So what is our job? To remove the viral nucleic acid material and throw it away. That is our job. So what we do, we will first try to grow thousands, billions of these viruses, right? And then we will do that. That can be done in egg. In egg, if you insert one virus, in chicken egg, millions of viruses will be made. Then you take those virus suspension and treat it with detergent, that is soapish material. What happens? That its membrane breaks and its nucleic acid material is also released. If you do heavy centrifugation, this nucleic acid material is relatively heavy. It will settle down at the bottom. And the broken membrane pieces will be here. You can remove them or you can treat them with enzyme and they will be all vanished. Then you take the supernatant, which is only membrane, and put it here. They're all broken because soap is there. Soap doesn't allow them to come together. And then you put the DNA. In that DNA, a circular DNA we can generate. Promoter is that of the udder gland, which is beta casein promoter of buffalo, which we isolated. And behind that, the pipe is that of human, let us say human gamma interferon, which is 14,000 rupees a vial. So there is a stitched construct where promoter is that of udder gland specific beta casein. And behind that, that pipe I have shown is that of human, that is human gene. And that's red in color. Now, when I got only surface membrane, this membrane has a speciality. They can fuse with human cell and puncture them and release whatever DNA they have inside. So now I mixed both of them. And as soon as I wash it with excess buffer and remove the soap material from it, what we call as soap, the alkalinity is gone. When we do that, these all viral membranes re-aggregate and try to make again a football. That's their property. But at this time, when they are making again a football, their nucleic acid is gone. But I have inserted human nucleic acid driven by promoter of beta casein of buffalo. That one particular piece, having both the things, will get inserted here. And when I remove the detergent, now you have a virosome. That was a virus because it has got nuclear material of virus. Now there is no nuclear material of virus, only the football membrane of virus, but my whatever DNA I want, I have inserted, that is human DNA, red colored. This particular thing is called as virosome. It is no more a virus. It's not infectious. It is not harmful at all. And now this, what we have checked that how it works, and this we have injected into the udder gland. And what we found that human gamma interferon, we found that when we take wild type females udder gland, there is no expression of human gamma interferon. But when we take those mice where we have injected with 75 microgram of the virosome or 100 microgram of virosome, the human gene is expressed. This is by RT-PCR, we could find out. That means we have successfully integrated the foreign gene into the udder gland. Then we have taken milk of such mice where we have done that job. You can do that. This is a non-lactating mice, you will not find any milk. And this is a lactating mice injected with the virosome. In this milk you can collect, or you can put them in buffer and can have more and more milk extracted. And then you try to measure human gamma interferon here, whether you really find in this milk human gamma interferon. And successfully we have shown that we can generate human gamma interferon in such animals as compared to the glands which were not injected. This will be a very good indication that we can generate human gamma interferon in the milk of a mice. Now we are going to the goat, that we will now have more milk in the goat. But this is a proof of principle, and this paper we have already published. Now we have got license from the government to work on goat and buffalo or cow to obtain large quantity of such therapeutics. This is not genetically modified organism. Society will not object to you that I don't want a full organism with a lot of DNA all over plate and not. No. Only udder gland is injected. And if you do not like such cow, you can just stop taking milk from it, and it will never propagate because its ovary or its testis doesn't have the gene of foreign interest. It's a very good technology which is coming up. So this is called as a bioreactor. So this cow or goat or buffalo can become my factory, and that will become, this is called as bioengineering. And some people call, because pharmaceuticals are produced in this way, so they call this biofarming also. And this is a very cheap way of generating therapeutic proteins in the milk, so that needful persons, even if they are poor, can really afford that in our country. Now I will bring you, change the gear, and I am just coming to the end of my talk. And I will say that in addition to that, we also work for something more important coming in our life. So let us say that there is a crisis going on with SARS-CoV-2. and we need to detect this. Now we are finding 4,500 rupees RT-PCR. So our institute has also worked where we have generated a very new technology. These are graphene rods, and these graphene electrodes, this is called as the N-COVID-19 antibody. So this is the antibody generated for the SARS-CoV virus surface protein. And on this graphene, we coat this antibody. And if in your nasal swab, the virus is there, then virus surface has the antigen. And this is that antigen which is present in the virus surface. So if you take a very sensitive technique where the antibody is coated, antibodies you can produce in tons by monoclonal antibody preparation. There are techniques. And you coat those in these graphene based electrodes. And when this COVID-19 surface protein or spike antigen comes in nostril swab, because it is present on the surface of those footballs, you throw it on this. If this attaches here, if there is a positive case, you will see there will be change in the reading in this machine. Because the current is passing from here to here, and the restriction is only by antibody. As soon as you add antigen, and if the patient is positive, antigen will bind here, and this will create resistance in the path of electricity. Immediately the voltage will change. And you can say this man is positive. So our target is that instead of 4,500 rupees PCR, this can be sold at somewhere around 150 to 200 rupees per test. So this we have generated. We have given it to a company who is really modifying a technology to equip this and attach it to a machine so that all results are coming into a printable format. So that's why we are there. So we do this kind of work also. Now, when all the antibodies are prepared, now new vaccines are coming for COVID-19. So what is going on? You read in paper, human clinical trials are going on with vaccines of Sputnik, of Serum Institute, of Bharat Biotech, and now Reddy's laboratory has taken Sputnik and Serum Institute in Pune. What will they do? They will use a portion of antigen of the COVID-19 causing virus, or they will kill the virus and make them dead and then inject into human beings. Human body will generate antibodies. Now, in human, you cannot challenge human with virus. You cannot through his mouth and nose give lot of virus and say, I gave you vaccine. Now I am giving you 100 ml of virus. I am seeing whether you will survive or die. Human beings will not allow that. So what's happening? That those antibodies, so I am, in addition to National Institute of Animal Biotechnology, I am also director of another institute as an additional charge in Delhi called as Translational Health Science Technology Institute. There we are doing that work that you cannot do experiment with human. So when you have given various vaccines, how do you test the vaccine is good or bad? How do you test the antibodies generated by that vaccine will stop the bad effect of the virus? You cannot do it in human. Human will say, give me injection, generate antibody in my body, take my 5 ml of blood, test it somewhere else. Don't give me virus. If your vaccine didn't work, I'll die. That's true. So you again need an animal model to test all such vaccines. So what we have done is that I'll give you an experiment. There is a very good model of hamster. Hamster behaves exactly like human for SARS-CoV-2 virus response. That means on day 1, before infection of hamster, you collect the blood samples and keep. That's a controlled blood sample. And then on day 0, you give virus exposure to the hamster, nose and mouth. On day 2, you check the spleen and blood. You can kill the animals. Out of the 20 animals, 5 you can kill here. And then you can, on day 2, you can do that. On day 7, you can do that. On day 14, you can do that. And see how the virus is affecting the body. And what they found exactly, I'll show you that when you give virus, these are the healthy animals. Up to day 14, they studied the hamster. In the virus given animal, these are the 2 groups of the virus, one little more, one little less. The body weight declined in all of them over a period of 15 days. This is the untreated animals. And you will find, if you compare the lungs, you will find the lungs of the treated animals gradually started becoming bad. Can you see the blackness in them? So this is what exactly is happening to human beings' lungs now. And by day 7, they are very bad. But by day 14, they are coming back to as good as the untreated animals, normals. Here we also see there is a heavy breathing distress. The animal perspires very heavily, just like human beings' problem, lack of oxygen. Now we have seen what happens to spleen. On day 2, as compared to untreated, spleen becomes little enlarged. But by day 4 and day 7, spleen of treated animals becomes very big as compared to untreated animals. But with the grace of God, by day 14, everything comes down and tries to become normalized as compared to control animal. So this is exactly what you are seeing. If you go, people will say 14 days quarantine. It's not a wrong idea. This mice also is giving you same kind of result. That by 14 days, the viral load goes up, and then it comes down by 14 days. So this hamster is a very good model. Hamster does not die, but it shows you all characters of lung pathogenicity. It shows you heavy breathing distress, and it shows that it comes back also. So now if I collect antibody of all the vaccines which are actually being done in this institution, all the people who are generating these vaccines, they are sending their serum of those human beings after vaccination. They are going to send us, and we are going to inject this into these hamsters. And we are going to see that if in these animals, those antibody of human from the serum is injected, or serum is injected to this hamster, can we prevent this pathogenesis of lungs? Can we prevent the breathing distress which occurs up to day 7 very heavily? And if we can do that, I think we have achieved what we wanted to achieve. So definitely this is a very good model, and this work is being done in another institute which is called as THSTI in New Delhi. So what I will end my talk by giving you few suggestions, that you have to be always think that your brain is to be revamped, to be refurnished, to be washed and clean every time. And something which garbage is getting collected, you have to throw out and put the good things in your brain. So for discarding and refreshing ideas, it is very important that you realize that you have to always sit calm and quiet and think what you should do. You should discard things which you do not need in your brain, for which what is needed. Flexibility of individual. You students should have immense amount of flexibility. That means that flexibility is very important, because your teacher is going to teach, your mentor is going to teach. But job of mentors is, we are managers. Our job is to force that whatever is needed in your brain, I will insert it. But your job is to remain flexible, get the ideas, listen to the seniors, listen to your teacher, and understand that this is very, very important. You to be very flexible for better utilization of the resources for serving humankind. So see, this is where I started my institute, where I came. This is just to motivate you. I am showing you. And within 2 years, I made this wonderful institution look like this. So nice. And not only that, now we are proud of this institution. And we work day and night to make this institute. This is just to encourage you that you can also be one of like me. You can also work very hard, grow very nicely in your life, and work very hard. You can achieve all these things and get all these credits in your life. So this is how nicely this institute looks. And God also favors, and this you will be more happy. This institute is under Ministry of Science and Technology. So you must be seeing Dr. Harshvardhan every day in TV. So he only inaugurated our institution on 11th of August 2018, and this is Secretary of Department of Biotechnology. So our institute was inaugurated by Dr. Harshvardhan, and we are really proud to be part of this Biotechnology Institute. And I will suggest you one thing: remain focused, like this woodpecker. Always, it does day and night labor to try to see whether it can make a house here or not. After 4 days, it may discover that this place is very hard and this tree is not for me. It will go to another tree, do the same job until unless it makes a house for it. So be very focused like this, very focused like this in life. I'm very sure lack of success or failure should not deter you. You should keep on doing this unless you really make a wonderful house for yourself. Never dodge. So psychologically, this guy, you love cricket. So I'm showing you this. Psychologically, he's prepared that whatever ball is going to come to me, I am going to get hit in the head. So even before the ball comes to him, he has turned his back. See, ball is going in other direction. So don't be afraid. Rather, jump towards the ball. Don't be afraid. See, he is also afraid. Ball has gone somewhere else. But look at this guy. He's jumping to catch the ball. He's giving his life to catch the ball. Even a person like me who is bulky can do this. He's not afraid of what loss he's going to incur. He's just focused and he's going to catch what he wants to do in his life. Right? So this is very, very important. I will just give you a say. Everybody wants happiness. Nobody wants pain. But you can't have a rainbow without a little rain. So life is plus and minus. You'll be sometimes happy, sometimes unhappy. Go through hardship so that it makes you a better individual. Keep your dreams very high. Keep several dreams. I'm very sure one of them will at least reach to the God and your dreams will be made true by Him. And with this, I will end my talk. And if you have any question, you can ask me. Thank you.
Thank you, sir. Thank you, sir, for your excellent, encouraging, thought-provoking talk. I am quite sure that still many of us in a spellbound condition after having this wonderful journey, virtual journey, though, in the world of the biotechnology. So thank you once again from our core of heart, our core of mind. Now, boys, it is now the session for the question. If you have any question in your mind, I think definitely you can ask it. So please try to start your question.
Sir, my name is. I have a question. You have said in your talk that the promoter region is tissue specific. Is there any exception to that?
1 second, I am trying to unshare. Is it the slides are still shown, isn't it?
Yes, sir, it is still showing.
Yeah. Let me see if I can. 1 second. Ravi. I'm not sure. Speakers. How to make quick? Vivek, sir. You're sure. Oh, now it has come. Sorry, sir. So I can stop sharing. Yeah. Now I'm fine. Yeah, you can ask me. Hello.
Hello, Tombok. Please ask your question again.
Yes, sir. My question is you have said in your talk that promoter region is tissue specific. Is there an exception to this?
Well, there are certain ubiquitous promoters. There are certain proteins which are produced in several parts of the body. Right. So they will be ubiquitous. Like ubiquitin is one of them, beta actin is one of them, GAPDH is one of them. There are certain proteins which are produced in several parts of the body. But if you want to really have a transgene which you want to produce in an organ, you can have a protein of that organ which is specific for that organ and isolate that promoter. Then the tagged gene will express only in that tissue. So it is not that all promoters are tissue specific. Where several promoters are tissue specific. So when you want to express a protein in brain, you don't have to take GAPDH or protein which is present everywhere. You take, let us say, glial fibrillary acidic proteins promoter. Tag any human gene, it will express there in the brain.
Okay, sir. Thank you.
So next question. So Oritronath, please ask your question. Please unmute yourself and ask the question.
Sir, how can we make an rat?
It's not naked rat, something is naked mouse is there, something which is called as nude mice. And it is actually deficient. Its thymus function is deficient. So it's available. Or you can really manipulate the thymus from childhood and can do that kind of job. And it loses its hair.
Sir, is it immunodeficient?
Yes, it is always immunodeficient.
Okay, sir.
So next one. Yes, Orijit Palit. So I am requesting Bodhisattva Mahara. Is there any question in the YouTube? Hello? Hello, Bodhisattva Mahara. Whether there are any question in YouTube?
YouTube. Just a minute.
স্যার কোশ্চেন আছে
Ask it, ask it.
বলো যদি বাংলাতেও জিজ্ঞাসা করতে পারো বেঙ্গলে।
স্যার ওই স্মার্ট ওয়ান কি করে ইউজ করতে হয় বলছো ওই ট্রান্সজেনিক অ্যানিমালের স্মার্ট ওয়ান প্রোটিনটা কেরাটিন দিয়ে কি করতে হয়
No, Smart one keratin the two are different. Smart one is a protein which is actually there is a gentleman, Dr. Soumitra Chatterjee, and he was in National Cell Science Center, Pune. Then he became director of Indian Institute of Chemical Biology, Kolkata. And it is his protein which he has discovered and found some role in Th1, Th2, immune action, and also in some carcinogenesis. And whenever we make that protein overexpressing, spleen enlarges. So with SMAR1, I was showing enlargement of the spleen. And keratin 14 is a promoter which is a promoter of skin. Keratin is a skin protein. And when you tag green fluorescence protein gene behind keratin 14 promoter, skin becomes greenish. That proves that transgenesis is occurring.
Okay.
That is just to prove that whatever we are doing, I say that I've isolated a promoter for skin. Somebody will say, prove it. So best way to prove it is give a visible color protein so that skin is showing green color. That's the best way to prove that your promoter is working only in skin.
আচ্ছা আর স্যার ওই ট্রান্সজেনিক মাইসের ওই মিল্কের ক্ষেত্রে আমরা ওই সেন্ডাই ভাইরাসটাই কেন ইউজ করলাম মানে অন্য ভাইরাস না ইউজ করলাম
Because earlier the papers are produced. This is another gentleman called as Devi Sarkar. He was director of IISER Mohali and otherwise he is a professor of biochemistry in New Delhi. He has published a paper in a very big journal called as PNAS, where he has shown that this virosome is relatively safe for treating certain cases of liver cancer, where you can insert the gene of interest into the live animals. So it's safe. That's why it is used. Nothing else. No other reason.
Okay. Thank you.
Sure.
Anybody else?
স্যার একটা কোশ্চেন আছে
Okay, please speak.
স্যার বলছি আপনি এক জায়গায় বলেছিলেন HBBX এক জিন বলে ওই জায়গাটা একটু বলবেন মানে ওই জিনটা কি আর কি
It is hepatitis B. So hepatitis B virus has several genes. Hepatitis B virus has several genes in its genome. There is one gene called as X, gene X. That portion is called as gene X. So if you cut that gene from the nucleic acid, whatever, the material of the virus, and paste it under liver promoter and make a transgenic mice, liver of transgenic mice will express that X gene of virus and immediately cancer of liver occurs. That's how you can have a model of mice with a cancer of liver.
Thank you, sir.
Anyone else?
স্যার আপনার পিপিটিটা কি একটু প্রোভাইড করতে পারবেন তাহলে খুব ভালো হতো
না পিপিটি গুলো আমরা প্রোভাইড কাউকে করতে পারি না কারণ এর মধ্যে অনেক ডাটা এখনো পাবলিশ হয়নি
আচ্ছা
উই কান আদারওয়াইজ আই উড হ্যাভ শেয়ারড কারণ এতে অনেক লোকের ইনভলভমেন্ট আছে যতক্ষণ না পেপার গুলো হয় ওইটা আমরা করতে পারি না
ওকে স্যার
So I am now asking Bodhisattva Maharaj whether there is any question in YouTube.
No, there are no questions on that front.
Sir, I have a question.
Rajuli, shall we wind up?
So Brotojit has a question, Maharaj.
Sir, how are the mice and hamsters are treated before giving them the viruses or some kind of before experimenting on them?
So there is something called as institutional ethics committee, and every animal house is registered with a central body of government of India called as CPCSA. That is Committee for the Purpose of Supervision of Experiments on Animals. That is in the Ministry of Animal Husbandry, and there are 6-7 scientists of country who are member of that. I am also one of them. That's a central body of government of India in Delhi. All the animal houses in country, whether it is industry or zoology department or big institutes of science, their animal houses are registered with that CPCSA. And CPCSA sends 1 or 2 of his local representatives in a particular city into the institute to make a body called as Institutional Animal Ethics Committee. All experiments before they are to be done has to be written in detail. How much anesthesia you will give to animal before conducting any experiment. If you are making a cut to the animal, how much antibiotic you will give, for how many days painkiller you will give, how you will give special care to the animal. These are all to be given in writing and signed. If they are incorrect, that Institutional Animal Ethics Committee consisting of about 7 people will make them correct or enforce that well-being of the animal is made. After that certificate is received, that certificate will go to animal house, and the in charge of the animal house, who is a veterinary person, will be present at the time of experiment, ensuring that all these steps are taken so that animal does not suffer any pain or any kind of stress. So it is all planned. It is all in the well-being of the animal that is taken care.
Sir, I have a
Any
question.
question yet remains?
Yeah, I had a question.
Yes. Ask it.
Sir, for specific gene transfer, when we transferring a human receptor to a mice, then is there any chance that this receptor can be treated as foreign particle in mice?
Foreign particles.
Yes, sir. When we are transferring the human receptor to mice for specific gene transfer, is there any chance that this receptor, human receptors, may be treated as a foreign particle in mice?
No. We do not inject it into blood of a mice. Anything can be treated as a foreign particle if it goes into any nose or ear or eyes, that is surface epithelium, or in the blood. But we don't do that. We insert it inside the cell.
Yeah.
Inside cell, there is no immunology.
Yes, sir.
It doesn't treat as foreign particle.
Mriganko, whether you are want to asking anything?
স্যার আমি যদি বাংলায় জিজ্ঞেস করি কোনো অসুবিধা আছে?
Yes, you can speak in Bengal.
বাংলায় জিজ্ঞেস করো, কোনো প্রবলেম নেই।
স্যার বলছি আপনি যেই ওই মাইসের ওই জায়গাটায় বলেছিলেন পোলিওর জায়গাটা, পোলিও রিসেপ্টর ইনসার্ট করে, স্যার সেই জায়গাটায় যে সবসময় যে ইনসার্ট করার পর যে যখন পোলিও দিয়ে আমি ইনফেক্ট করছি তখন সে পোলিওর মতনই কোনো ডিজিজ ক্যারেক্টারিস্টিক দেখাবে, তার কি কোনো মানে আছে?
শুধু মাইসে ঢাললে তো দেখাবে না, দেখায় না। কিন্তু যে মাইসে তুমি,
আপনি যখন,
হ্যাঁ বলো।
বলছি রিসেপ্টরটা ইনসার্ট করার পর সে যে পোলিওর মতনই যে ডিজিজ এফেক্টটা দেখাবে, তার কি মানে সেটাই দেখাবে এরকম স্পেসিফিক কোনো রুল আছে?
না, এমনি কিছু রুল নেই। কেননা পোলিও যদি 10 জনের হয় তো সবাই এক্সপ্রেস করে না সিমটম। বডির স্ট্রাকচার, তার ইমিউন সিস্টেম, তার উপরে ডিপেন্ড করে। যে পোলিও তো অনেক মানুষই হয়তো এক্সপোজ হয়, সবার পোলিও তো হয় না। সেমনি মাইসেও যদি তুমি 10টা মাইসকে পোলিও, মাইলাইটিস, ট্রান্সজেনিক মাইসকে পোলিওর ভাইরাস দাও, হয়তো 3টে মাইসেও সিমটম দেখা যাবে, তাদের নিয়ে কাজ করতে হবে।
আচ্ছা, তাহলে
এই
সেই
কিছু সার্স কোভিড-19 তো তাই, সবার কি একরকম হচ্ছে, রিয়াকশন একরকম হচ্ছে?
স্যার আর একখানা,
বলো বলো।
হ্যালো
হ্যাঁ বলো
হ্যালো
বলো,
স্যার,
শোনা
হ্যাঁ
যাচ্ছে।
স্যার, আর একখানা কোয়েশ্চেন আছে। আপনি এই যে কোভিড কিট ডেভেলপ করার জন্য এই যে গ্রাফিন ইউজ করেছেন, স্যার গ্রাফিনের আশেপাশে মানে ইলেকট্রনিক কন্ডাক্টিভিটি ডিপেন্ড করে, আমি যদি জিঙ্ককে দেখি বা ম্যাঙ্গানিজকে দেখি বা গ্যালিয়ামকে দেখি, এইগুলিরও তো অনেকটা সেম কন্ডাক্টিভিটি, তাহলে এইগুলিকে কেন নিলেন না?
গ্রাফিন বেস্ট, লোকেরা যারা এই ফিল্ডে আছে, যারা সেন্সর বায়োসেন্সর বানায়, তারা অলরেডি টেস্ট করে দেখেছে গ্রাফিন ইজ দ্য বেস্ট অ্যান্ড চিপেস্ট। এ কস্ট ইজ অলসো ইনভলভ, মনে রাখবে। একটা কিট বানাতে গেলে কার বেশি কন্ডাক্টিভিটি, তার দাম যদি বেশি হয়ে যায় তখন মুশকিল। রূপোর কানের দুল দিয়ে যদি কাজ হয়ে যায়, তাহলে সোনার কানের দুল পরার কোনো মানে নেই।
Network problem.
Okay. So I think now it is time for the vote of thanks. So for that, I am now requesting Swami Vedananda Ramdaji Maharaj for the vote of thanks.
Respected Dr. Subeer Majumdar, Director, National Institute of Animal Biotechnology, Hyderabad. So we are very grateful to you, sir, for your wonderful talk this evening as an induction talk for the newly admitted PG students, for the newly admitted M.Sc students in Zoology. So we are really indebted to you, sir, for your beautiful talk on various new novel areas like germ cell transplantation and non-GMO experimentations and various other areas which you have highlighted. We are looking forward to you, sir, for again getting enlightenment from you. Whenever the situation improves, please visit our college, and
Definitely.
whenever you come to Kolkata, we'll expect that we'll try to be in touch with you, sir. And in future also, if possible, please accept some trainees from our college in your lab for some brief projects or summer internships or whatever possibility is there, so that hands-on training from you, sir. So we are very much happy and glad to have sir amidst us, and we express our heartfelt gratitude on behalf of the department, on behalf of the college, for this beautiful induction program talk delivered by Dr. Subeer Majumdar, Director, National Institute of Animal Biology, Hyderabad, and on the topic life, genes, biotechnology, and needs of human. So thank you very much, sir, and thank you all teachers and all for actively participating in this induction program. Hope that it has ignited our endeavors, our interests, and we will all try to work hard to make academics research much more lively and much more in depth. Thank you very much, sir, for your for giving us so much time from your busy schedule and enlightening us. We look forward to get in touch with you. Please visit us and please be in touch with us so that we can be in your warmth of your knowledge and your concern. Thank you very much, sir.
Thank you. Thanks a lot. Thank you.
যাক সুবীরবাবু, আসতে হবে কলকাতায় এলে আমাদের কলেজ ভিজিট করবেন।
নিশ্চয়ই নিশ্চয়ই, ডেফিনেটলি। ভালো
হ্যাঁ
থাকবেন।
ঠিক, হ্যাঁ আপনারাও ভালো থাকুন, এই আর কি, ভালো থাকবেন।
থ্যাংক ইউ, টেক
হ্যাঁ
কেয়ার।
সব, হ্যাঁ হ্যাঁ, ভালো থাকবেন। নমস্কার নমস্কার। ব্যাস, তাহলে লিভ করে যাই, সবাই লিভ করে ফেলা।